Um, all right, so my name is Brandon Durant. I'm a PhD student at the University of Buffalo, and today I'll be sharing some of my research, which is entitled Investigating Spatiotemporal Dynamics in Microbial Community Diversity and Composition Within Surface and Groundwaters at an Urbanized Beach with Impaired Water Quality. So we're going to start out with some of the background. So hmm, it's a bit difficult because I'm like, if the slide doesn't change with the words, well, to me it's changing, but I don't know if you all can see it change. So it might have you all more lost.
It's changed. You're good.
Okay. Uh, so we're going to talk about bacterial pollution within recreational freshwaters first. Bacterial pollution of recreational freshwaters is a growing problem that has socioeconomic and public health implications. When bacterial concentrations in recreational waters gets too high, beaches are closed to the public in order to protect human health since water exposure could result in illness. To protect human health and prevent illness, the EPA has developed and enforced water quality standards that are based on the cultivation and enumeration of fecal indicator organisms, E. coli and Enterococcus. Despite not being pathogenic themselves, meaning disease causing, these indicator organisms, which behave similarly to pathogens, are used to estimate the levels of fecal contamination and hence the risk associated with water contact um, within recreational waters. Although this provides important water quality information, no information regarding the actual source of pollution itself is provided, which makes mitigation efforts very challenging. Analysis of microbial communities using next generation sequencing technologies has provided us with the alternative and more informative ways to assess water quality in recreational waters. Instead of the previous approach, where we take a water sample and use it to culture and enumerate the fecal indicator organisms that we just talked about, with this approach, we instead take our sample and extract the DNA from the bacteria, and then we amplify the DNA, get it sequenced, and then analyze the resulting sequencing data. So using this technology lets us identify key bacterial community members and determine the specific roles that they play within their environment, which is extremely useful when tracking bacterial sources and understanding the impact that certain bacteria may have within recreational waters. If it's if there's too much of a disconnect, just let me know. I'll slow down or anything. So this technology has been used um, in many studies, and it's allowed for the identification of fecal inputs into polluted water bodies in numerous aquatic environments and helped researchers gain a better understanding of the spatial and temporal dynamics of microbial communities worldwide. Here at UB, with a previous study that I've done, we also use this technology to investigate bacteria at Woodlawn Beach, which is our very own local beach, which I'll just share with you briefly. The goal of the study was to investigate the spatial and temporal dynamics of microbial communities at Woodlawn Beach and see how they compared to three other nearby beaches. So, Woodlawn Beach is a popular beach on the eastern shore of Lake Erie that usually opens during the summer when the weather gets warmer. Unfortunately, because of its high E. coli levels, it's believed to be facing a contamination issue, and as a result, is closed for a significant portion of the swimming season. So we collected water samples from uh, Bennett Beach, Gallagher Beach, Hamburg Beach, and Woodlawn Beach. Those are the four Eastern Lake Erie beaches that I mentioned before. And then at Woodlawn Beach, we collected surface and groundwaters. and then we analyzed them. And what we found was that the microbial communities among the 4 beaches showed significant spatial and temporal differences over the 4 weeks of sampling. Then at Woodlawn Beach, surface water microbial communities were different from groundwater microbial communities. And the microbial community at Woodlawn Beach, the actual swimming water itself, was more similar to the surface waters rather than the groundwaters. And finally, we didn't really see any significant sewage-derived contamination within the swimming waters at Woodlawn Beach. So while going through the literature that was relevant for this research, one thing that really stood out was that there wasn't a lot of work done on groundwater microbial communities. So we kind of saw this as an opportunity, and thought it would be interesting to see how the composition and the dynamics of surface water bacteria compared to groundwater bacteria. So that leads us to our next study, which is the main topic for today. And we'll just start with some of the background. So surface and groundwaters represent 2 very different but hydrologically interconnected, biologically active environments. Because of this connectivity, microbial communities in surface waters have the potential to interact with those that are in groundwaters. And this has implications for both human and ecosystem health within recreational coastal waters, which receive inputs from both surface and groundwater environments. To date, research performed within groundwater and ecosystems has focused mainly on changes to microbial community diversity, structure, and function associated with contaminated aquifers. Contamination by various compounds and chemicals, such as arsenic shown here, were found to reduce microbial community diversity and alter community composition. Additionally, while investigating gene diversity and functionality within contaminated aquifers, he et al. Found that functional diversity was actually a pretty good, functional gene diversity was actually a pretty good indicator of groundwater contamination. Other studies that were done in non-contaminated aquifers have investigated microbial community responses to specific events such as stratification and seasonal change. Studies that considered the spatiotemporal dynamics within groundwaters have mostly found that in addition to being spatially unique, microbial communities were found to be both spatially and temporally stable. Despite all of the research that's been done, however, within surface water systems and the recent work done within groundwater ecosystems, not many studies have focused on both of them at the same time. Of the few that have, there's been a big focus on surface groundwater mixing zones due to seawater intrusion. Microbial communities within these intrusion zones were usually significantly impacted by seawater intrusion, and that could be seen by the change in community composition between high salinity and low salinity samples. There was also another study that looked at how flooding events impacted microbial communities in groundwaters also. But although these connections were made between surface and groundwaters, there hasn't really been any studies on surface and groundwater communities to determine how the interaction between them could influence recreational coastal water quality. So that brings us to the main topic for today, which is investigating spatiotemporal dynamics in microbial community diversity and composition within surface and groundwaters at an urbanized beach with impaired water quality. The specific research tasks that we wanted to address are: number 1, investigate the spatial and temporal variability in diversity and composition for microbial communities in surface and groundwaters at different depths. The second one, identify predominant and differentially abundant taxa, which explain the spatiotemporal differences observed between surface and groundwater microbial communities at different depths. And then the last one is to determine whether dominant taxa within surface and groundwaters at different depths correlate with measured environmental variables. So at Woodlawn Beach, we collected both surface and groundwater samples. Surface water samples were taken from LE2, which is down here, and that's the swimming water by the lifeguarding station. And then they were also taken from Blaisdell Creek and Rush Creek. This is upstream on Rush Creek, where both Blaisdell and Rush converge and then flow into Lake Erie just north of LE2, of the lifeguarding station. We also collected groundwater samples. So for groundwater samples, 5 monitoring wells were drilled at different locations into an unconfined aquifer, which encompassed our area of interest. Each well included both a shallow well, which was screened across the water table 15 feet deep, and a deeper well, which was installed at the bedrock surface. So after we collected our samples, we prepped them for sequencing, sent them in to be sequenced, did some quality control on the returned sequences that we got, the raw sequences, and then we just analyzed our data. So we're gonna go back to our first research task, and I'll show you some of what we found there, which was looking at the spatial and temporal variability within diversity and composition for microbial communities in our surface and groundwaters. All right, so let's start with diversity. Shannon-Weaver alpha diversity is what we're referring to here. It's like a metric of community diversity, but what exactly is that? So to start, we're going to go over a couple definitions, so you know the differences between them. And the first term here that we need to know is abundance. So abundance here refers to the total number of organisms in an area, and it does not matter what species they are. Species richness refers to the number of different species in an area, and it does not matter how abundant they are. Diversity incorporates both the number of species in an area and the evenness of their abundances. So let's look at what that means. Let's say we have a community of birds, and we decide to count them. So here in community A, we see that we have 10 birds, right? So that means that our abundance here for this community is 10. So next we observe that out of these 10 birds, there are, although some of them are repeated, we only actually have 4 different types of birds, which are indicated by the 4 different colors here. So our richness is 4. Finally, we're going to go into diversity. For now, I'm going to leave that as a question mark, and we'll come back to that later. So if we compare community A to community B, we see here that we have the same abundance of 10 and the same richness of 4. However, you also notice that this community is a bit more balanced in terms of their the distribution of the 4 birds. So you would think that the diversity in community B is probably higher than the diversity in community A. This is where our diversity indices that tell us what the diversity is, this is where that comes in. So diversity indices calculate community diversity by accounting for both the abundance and the richness that we just spoke about of each species within the communities. So here we see that even though we have the same abundance and richness within each community, the differences in relative abundance of each individual species within these 2 communities means that the 2 communities will have different diversities. However, there are also multiple kinds of alpha diversity indices. So I'll just talk about 2 of them that are used frequently are the Shannon Diversity Index and the Simpson Diversity Index. The Shannon-Weaver Alpha Diversity Index is the one that we're going to be using for our research. But then there's another one just to show you the difference between different diversity indices. So the main difference between these two, the Shannon index emphasizes rarer species when it calculates diversity, and then the Simpson index it emphasizes dominance by one species. So in this case, if we use the Simpson index to calculate our community diversity instead, it would show that the diversity within community A was higher, since community A is dominated by one species. So again, the first thing we want to do here is determine if alpha diversity between surface waters, shallow groundwaters, and deep groundwaters were significantly different or not. And on the axis here, when we're looking at this graph, we have our sampling locations, with the first 3 being surface water sampling locations, the next 4 being shallow groundwater sampling locations, and then the final 5 being deep groundwater sampling locations. On our y-axis, we have the Shannon-Weaver Alpha Diversity Index that's used to calculate community diversity. And then each of the dots that you see here, it's a different microbial community, a different sample that was used to a different microbial community within a different unique sample. And to find out if they were significantly different or not, we used an ANOVA test. I didn't show the results here, but I'm only showing the significant results. So we compared the diversity between these 3, and to see if they were significantly different, both temporally and spatially. And we found 2 things. The first was that the microbial communities between surface waters, shallow groundwaters, and deep groundwaters were not temporally different between the 6 collection dates, which I didn't show that here. And then the other was that although the alpha diversity between surface waters and groundwaters, although that was not significantly different, alpha diversity between shallow and deep groundwaters was significantly different. One thing that was also very interesting to observe was that within our shallow groundwater community here, you see this one sample that had consistently low alpha diversity. That was MW3S, the shallow well within the third, the shallow depth in the third monitoring well, yeah. And its diversity seemed to be extremely low compared to the other shallow groundwater wells, which it's just not characteristic of the shallow groundwaters. So that was interesting to see here. But to continue our analysis, our spatial analysis at least, we looked at spatial variability between surface waters, shallow groundwaters, and deep groundwaters using a Bray-Curtis NMDS plot. So what exactly is that? So a Bray-Curtis non-parametric multidimensional scaling plot, or NMDS plot, is a distance metric that is based on both richness and abundance, which we just talked about. And it's used to visualize how similar or dissimilar samples are in relation to each other. So on these plots, samples that are more similar to each other will plot closer together, and samples that are more dissimilar will be farther apart. The observed distances that we see here are all relative, so the axes don't mean anything. So to determine if the groupings we see here are statistically significant, we use an Adonis test. And the null hypothesis for this is that microbial communities between our defined groupings are the same. So if we get a significant result, less than 0.05, then that says that our communities are actually different. Also, with these graphs, the stress, the value that you see here, it informs us of the usability of our data, with any stress under 0.2 being acceptable. So now that we know what that is, we visualized our microbial communities using this plot, this visualization, and each of the dots here was a different microbial community or sample. So this was done for surface waters, and then also for shallow groundwaters and deep groundwaters. And what we found here was that microbial communities were not similar between the three sampling depths. Surface water samples were significantly different from the groundwater ones. Also, I didn't show it here, but the microbial communities were not temporally different over the 6 sampling dates, just like with the diversity. So going back to the spatial analysis, we can also move from looking at this broader picture, like all surface and groundwaters, and to just go to looking at the like the groundwater community by itself. And when we do that, what we find is that the microbial communities within shallow groundwater samples were significantly different from those within the deep groundwater samples. Here you see there's the blue circle. That's just all of the deep groundwater samples, and they are kind of separated out from all of the shallow groundwater samples. So we're able to see these differences again between surface and groundwaters. Then within the groundwaters themselves, we can see that as well. But then what's even cool is that you can keep going down in scale. So we can even see the differences within each of the sampling depths. So here we see site-specific microbial communities as well. So for example, if we look at the shallow groundwater microbial communities, we can see site-specific, spatially distinct communities between our different shallow monitoring wells. What's also interesting to note here is that if we look at the groundwaters versus the surface water communities, we see that the groundwater communities are a lot more distinct than when we compare them to surface water communities, which seem to be more spread out. And again, none of the communities among the 3 depths were temporally distinct, even though surface waters were kind of close. And in line with what we had done before with the diversity, I also wanted to point out when we look at this overall visualization of the surface and groundwaters, again here I see that MW3S kind of separated itself from the other groundwater samples, so that tells us that it's different from the other samples, which was very interesting given that we had also seen that it had uncharacteristically low diversity for being in that shallow groundwater set. So to recap, for this first research task, what we saw was that microbial communities showed spatial but not temporal variability in alpha diversity and composition among surface waters, shallow groundwaters, and deep groundwaters. So our next task here is to identify predominant and differentially abundant taxa, which explain the spatiotemporal differences observed between surface and groundwater communities at different depths. So to complete this task, the first thing that we did was identify the most abundant members within our surface water, shallow groundwater, and deep groundwater microbial communities. So here we're just showing the distribution by sampling location of the most abundant phyla within surface and groundwater microbial communities. So any of the bacterial phyla that showed up at an abundance of 1% or greater within any of the sites, not the depths, but within any of the sites that make up those depths. If a phyla is at 1% or higher within any of those, we included those within this analysis. So what we saw was that regardless of sampling depth, the Bacteroidetes and the Proteobacteria phyla were observed. Proteobacteria being blue, Bacteroidetes being brown. So these were kind of ubiquitous phyla, regardless of the environment. These are phyla that we saw. Within surface waters, the actinobacteria and cyanobacteria were observed at higher abundances. Within our shallow groundwaters, the OD1 and OP3 phyla were observed at higher abundances. And then the Firmicutes phylum was seen at higher abundances within deep groundwater samples. But interestingly also, it was observed at a higher abundance within MW3S as well. So the next thing that we wanted to do here was identify some of the differentially abundant members within our communities that might help explain some of the differences that we have seen so far in diversity and composition. That might help us, yeah. So this is important because it can inform us about, for example, key community members that are responsible for nutrient cycling under various environmental conditions. For now, this is just here to show that the work has been done. I'm not going to sit here and name off a whole bunch of bacterial names to you all, as they don't really mean much right now. So this is here mainly just to show that this was done. So yeah. But also, we did this for just the shallow groundwater microbial communities. Because of those big differences that we saw between MW3S, the third shallow monitoring well, because of the differences we saw between that and the remaining shallow monitoring well microbial communities, we also wanted to see why, or get like to the bottom of why those differences are, why it's happening, like why they exist. So again, this is here for that. So just to recap, for this research task, what we found was that differentially predominant phyla were observed within surface waters, shallow groundwaters, and deep groundwaters. Yeah. So moving on to our final research task, which is to determine whether dominant taxa in surface and groundwaters at different depths correlate with measured environmental variables. So we collected some basic water quality information that kind of provides us with water quality context. So the first thing we did was we analyzed this data, and then with that data and the sequencing data that we had that I previously gone over, like the Bray Curtis, we put them together, and we wanted to see how our environmental parameters compared between our three depths. So we're just going to go through and kind of construct one of these plots together so you know what's going on when we review what we've done with this. So here we just begin with a blank plot. And then we add our surface water communities, which are represented as white dots, followed by our shallow groundwater communities, which are represented as gray dots. And then finally, our deep groundwater microbial communities, which are represented as black dots. So on these plots, we then visualize a specific environmental parameter. In this case, for example, alkalinity. And then this visualization is done using contour lines. So the range of values for the specified environmental variable is shown right under the name of the variable. And then finally, we include a REML score, which is a maximum likelihood statistical test, which informs us of the goodness of fit of our environmental contours. So this is for alkalinity, but if you look on the side first, is my mouse here? There we go. So if you look on the side first, next to these graphs that we just made, you'll notice that there's a plus sign, a minus sign, and then there are bacterial phyla under that. This is just me reporting the positive correlations and the negative correlations. which were determined using Pearson's correlation at a 0.05 cutoff, significance cutoff, between the predominant phyla and the environmental variable for each of the three depths. But this isn't global, which is part of what we're going to continue to do. So it's not all three depths at the same time. It's looking at surface waters, looking at shallow groundwaters, looking at deep groundwaters. So it's not a global thing. It's just right now what I'm presenting is just it's depth specific. So for example, I'm not going to go through all these. What it says is that within surface waters, alkalinity is positively correlated with bacteroidetes. Within surface waters, alkalinity is negatively correlated with the varicomicrobia, cyanobacteria, plantomycetes, and chloroflexi phyla. I'm not going to go through all these names. Again, it's just right now we're looking up exactly, like specifically what each of these phyla do, what are some of their predominant members, how they should respond to certain things to kind of clear up that picture. But I just wanted you to know what was done and just so you can follow as well. So we're going to start with alkalinity since we're already here. Alkalinity, ability of water to resist changes in pH. What we saw from our data was that alkalinity was lowest in surface waters and observed at higher levels in both shallow and deep groundwater samples. We also did pH. If we have alkalinity, we require pH to kind of make sense of that data. So ability to resist change in pH. If you don't have the pH itself, then it's not really it could be useful, but you usually use both of them in order to get a better understanding of what's happening. So pH is how acidic or basic the water is. And certain organisms, certain bacteria can thrive at certain pHs, but then other pHs would kill them. The same for any organism, really. So this is one of the more important parameters. And what we saw was that, for the most part, pH was kind of at the same level among all surface shallow groundwater and deep groundwaters. It's just at that MW3S sample that we saw a spike in pH. But other than that, and also I didn't show it here, but when you take out that MW3S sample, pH is no longer significantly different between these three sampling depths. So next we went on to dissolved phosphorus. Both phosphorus and nitrogen are nutrients that are known to generally control eutrophication within surface waters. So dissolved phosphorus we saw it at lower levels in surface waters, higher levels in deep groundwaters. For dissolved nitrogen, on the other hand, was the exact same thing, lower in surface waters, higher in deep groundwaters. Next was ammonium. It was the same trend as it's one of the reduced forms of nitrogen. It was same trend as the nitrogen as well. It was observed at lower levels in surface waters and higher levels in deep groundwaters. For chloride, since we're not near an ocean, we're in an urban area and we're not near an ocean. This is Lake Erie, which is freshwater. So usually, if you have chloride, I think it's in groundwaters. It's more because of anthropogenic inputs like wastewater or load salting or something. All of this is part of the work that we're doing right now, trying to get into the nitty gritty of what we should see, what we see here, what does that mean, what are the specific bacteria doing. But for chloride, we saw it at higher levels in surface waters and deep groundwaters, and at lower levels within these shallow groundwaters. We also looked at dissolved organic carbon, and that was seen at lower levels within surface waters and higher levels in groundwaters, which usually you would think it would be extremely low in groundwaters and higher in surface waters. But that's not what we saw. So what are these? They get up pretty high, 7-8 milligrams per liter, where it should be like under 4, like 1 to 2. We also looked at dissolved oxygen. This could give us an idea as if the water is from recent recharge, or if it's older water. It also tells us about what bacterial members we should be seeing there. And we saw this at lower levels in groundwater, higher levels in surface waters. And we also looked at specific conductivity, lower in surface water, higher in groundwaters, and sulfate, which was seen in relatively similar levels across the 3 depths. However, again, there was a spike in MW in the third shallow monitoring well. However, unlike pH, when you remove MW3S from this analysis, which I didn't show here, it's still significantly different. I'm showing what we observe here. We also have statistical tests that I think it's in the appendix. So for this last research task, what we found was that there were significant correlations between some of the predominant taxa and the measured environmental variables. Within the surface water, shallow groundwaters, and deep groundwater microbial communities. And here I'm just showing the research conclusions that we came to. Of course, additional work is regarding the role that some of these phyla have within especially the groundwaters. The continued work would be finding out what they're doing there, what we should be seeing, what does it mean, like just getting into that type of stuff. So that's what we're actually doing right now. Why is MW3S so different? So yeah, that's the work that I've done so far. Anybody got any questions? Hopefully the timing with the slide switching and the audio was fine.
Thank you so much, Brandon. I'm curious if you can kind of sum it up for us, if there's any sort of a conclusion that can be made with the beach water quality and your research.
So me personally, this is a bad phrase to use, but I don't want to jump to a conclusion and just say, oh yeah, this and that to look smart in front of you guys, because that's specifically what I'm doing right now. Right now, talking to some of the professors on my committee, what we agree on is that this isn't what we see so far is not characteristic of a pristine, uncontaminated aquifer. I'll say that. But again, there's still more things that I need to research before I would just conclude something right now. As far as like actual beach water quality itself goes, one thing I will say is that before, with the culture-based methods, where they assumed that these fecal indicator organisms were the only place they could come from was from a gut flora, so it had to be within an organism in order to get into the water, so it was indicative of fecal contamination. There's a lot more studies that have been done that show that these fecal indicator organisms can actually thrive and potentially replicate within the natural environment. So that's why there's been a lot more research in trying to find alternative indicators that might be better than what was previously assumed. So yeah, that's what I'd have to say right now. But I would definitely have to do a little bit more research first before I conclusively said, oh, we're screwed, like don't go in the water, something like that.
Hey, Brandon.
Mm-hmm.
These groundwater wells, they're fairly shallow, aren't they? The bedrock's fairly shallow at that point?
I'm sorry?
The deep groundwater wells, they're fairly shallow, aren't they? The bedrock is fairly shallow in that location, is it? How much deeper are the deep wells than the shallow wells?
Shallow wells were 15 feet deep. And then the deep ones were they were variable. They were at differing levels. I have the numbers. I don't have them memorized. Yeah, I have the numbers, but I don't have them memorized. They were fairly shallow though. Yes. Yeah.
Yeah, there's not much difference between them. I did notice that there was peat in the geologic profile of some of those wells, which may explain the organic carbon.
Yeah. Also, we as we're still in the process of looking it up, but that monitoring well, the third monitoring well, there's a steel plant that's nearby,
Yeah,
and the spike in pH got up to like above 11. So we're like, oh, maybe it's because of that that there might be a spike. What is it called? Slag?
yeah, right.
Yeah. But we're still looking into all this. I'm literally looking up like how a specific type of soil will affect the microbial community. Uh, what is a reason that pH might be super high? Things like that. Just everything that we need to know about this. That's what we're currently working on right now, which is why it's like, okay, I can't just conclusively say this and that.
Great. Anybody have any questions for Brandon?
Nope, I have none.
Okay, not hearing anything. Thank you, Brandon. That was very interesting.
Thank you.
Good luck with the research. Thanks for joining us.
All right.
Okay, we can go ahead and move